Hi everyone, I'm Jennifer Sun. I have the honor of serving with Tom Gardner as the co-scientific directors for the MTM Vision Initiative. And we put Ryan up first, front and center, just like last year, because he speaks so movingly about his experience and because the experience of our patients, our loved ones with diabetes, is really at the heart and center of the work that we do. So I'm going to talk now about some of the science that has grown out of this, but again, I think one of the things that makes this initiative incredibly special is the passion that we feel for the mission. So I have the pleasure of talking about some of the clinical studies that we've developed over the last year, coming off of the work that was performed at the workshop last year. Here are my financial disclosures. And I want to start by acknowledging, as Robert did, the working groups that participated in our diabetic retinal disease staging system update effort. These are the leadership of the groups in each of those 6 working areas of focus, and they did a tremendous job and an enormous amount of work putting together targeted narrative reviews over the course of a couple of years to really comb the literature and their knowledge to understand what were the priority variables that might rise to the top as we thought about expanding our current staging systems for DRD into a more multidimensional and holistic system. And the beautiful thing about the work, although it was very clear out of all these groups that there is a great deal of work left to do to understand and validate and better characterize essentially all of these variables, they left us with a fertile ground of priority variables, and we had them target them to different levels of diabetic retinal disease severity, subclinical, early stage, mid-stage, and late-stage disease, and also to identify which variables were potentially ready for current use or within the next 1 to 2 years, which ones had a longer time horizon with unmet research needs that could be defined or that were promising in a potential way but might need a longer than 5-year time horizon to bring to prime time. And so these priority variables, many of these were biomarkers that we have drawn from to now move into that third plank of the first phase of our very ambitious program to look at new endpoints for DRD research and clinical care. And so I'm going to start with a slide just talking about the definitions of biomarkers and endpoints, because many of us, will use these terms, and I would just want to make sure we're all on the same page. Biomarkers, as defined by the FDA NIH Biomarker Working Group in their best resource, are characteristics that are objectively measured and evaluated. And they can be an indicator of normal processes, pathogenic processes, or of treatment response. And it is our hope to translate some of these biomarkers discussed by our working groups into surrogate endpoints if we can perform studies that show that they predict clinical benefit. In addition, some of the things that we talked about in the visual function endpoints might be clinical endpoints that directly reflect how patients feel, function, or survive. And both surrogate endpoints and clinical endpoints, the FDA and NIH groups have made very clear, continue to need validation for specific contexts of use in the setting of treatment, in the setting of monitoring, in the setting of screening, et cetera, to be used as primary endpoints for registration trials. So it's this work that we're about to, or we have been embarking on actually really over the last year, and that we will need all of your help with. Is there a need for new endpoints in DRD? Well, we think there is. With scientific advancement, we have the ability these days to affect a much wider variety of targets and mechanisms in DRD than ever before. And many of these are upstream of what were originally characterized very late stages in the disease severity. Furthermore, as our standard therapies for late-stage disease improve, and we've got some very effective therapies on hand now, it becomes harder to meet our current thresholds for efficacy. You might ask the question, well, perhaps we don't need new thresholds for efficacy if the therapies we have are already effective. But the fact is, new endpoints remain a priority for us because our current therapies do not eliminate vision loss by any means. We have non-responders, we have incomplete responders, and our therapies also have patient and societal burdens in terms of cost, inconvenience, and risks. So there is a need for new surrogate endpoints that address upstream mechanisms and early-stage diabetic retinal disease that can be shown to reliably predict clinical benefit. We also need additional clinical primary endpoints to describe the impact of DRD on aspects of vision and patient function that may not be currently addressed. We measure vision every day on our charts with black letters on a white screen, and it's very high contrast, and it really doesn't address how patients function in their day-to-day lives. So, major goals of this effort, the primary goal is really to work towards the eventual development of new recognized primary endpoints that may be acceptable for use in clinical trials for regulatory approval. That is a high hurdle. So we have very important secondary goals as well. And we think the throw-offs of this process may be the validation of surrogate or clinical endpoints that can still be used to enhance research or clinical care, even if they're not suitable for registration trials. And a great example of this is central retinal thickness on optical coherence tomography. It's not well correlated enough with visual acuity outcomes to ever be acceptable as a registration endpoint, and yet we use it every day in our clinical care and in our research to look at proof of concept and early efficacy. We recognize that this validation process will not stop and end with these two studies that I'm going to discuss. It will require probably multiple prospective studies, a collaboration across our community with diverse cohorts and defining specific contexts of use. And Robert mentioned these assessments of key test probabilities, and to us, this is really what that development, validation, qualification process for these variables entails. Really a better understanding before they get put into these incredibly long-term and expensive phase 3 trials, an understanding of the tests, of their variability, of their normative data availability, of their floor-to-ceiling effects, the degree of subjectivity in testing, the differences in how they change between eyes that are healthy and eyes with any DRD or late stage or different stages of DRD, so that we know what it means to have a real change on these tests. Without understanding these characteristics, we can't tell you whether a change that occurs over the course of a clinical study is meaningful or relevant. Of course, feasibility concerns, device availability and cost, clinical relevance and patient acceptance and time requirement are going to be important. So Robert talked a little bit about our progress already. We've made a lot over the last year since we first held our workshop in person here in October of last year. We've drafted the first 2 study protocols, and I have to give a shout out to Maureen Maguire, who's here, and also to Danny Liu, who's a biostatistician at the Jaeb Coordinating Center, who have spent an enormous amount of time and effort to help us draft these protocols and work through many of the challenging initial discussions. I'd also like to thank everybody who participated in the ARVO sessions and the webinars. We drew from the community not just of diabetes researchers, but inherited retinal disease, age-related macular degeneration, and we had participants from industry, academia, patient advocates, et cetera. So a proposal for collaboration on these 2 studies has been presented to DRCR Retina Network. For those of you who are not familiar with the network, I actually serve as chair for the diabetes studies of this group. It's an NIH, primarily NEI-sponsored consortium of sites that's performing clinical research in retinal disease. We've received funding from NIDDK, from JDRF, from other foundations, from industry partners over the years. And we have approximately 130 active sites across the US and Canada. So we're a standing infrastructure of sites ready and available to do research in retinal disease. And we get very excited about innovative new studies and partnering with other collaborations. Over the last 20 years, we've performed probably over 30 studies in diabetic retinal disease, and I'm proud to say that I think much of the way we treat diabetic retinopathy and diabetic macular edema across the world has been informed by many of these studies. So we proposed 2 studies. One is a study of the natural history of changes across natural history of retinal function in eyes of patients with diabetes, and the second is a study of eyes that are just starting treatment with intravitreal injections of anti-VEGF medications for diabetic macular edema or swelling of the central retina. These are multi-center, longitudinal observational studies, and the primary goal is to really define the ocular, structural, and functional characteristics of people with diabetes, covering a very broad range of diabetes duration and disease severity, both over the natural history of the disease and under treatment. Secondary goals that are important: investigating retinal structure function relationships, the degree to which measures change with increasing severity of DRD, test-retest variability, correlation in test characteristics between 2 eyes of a single individual, and finally understanding whether these both structural and functional measurements can be validated as surrogate outcomes or clinical endpoints. We'll be enrolling, for now, adults with diabetes. In the natural history study, we may also include non-diabetic control participants. The natural history study will enroll patients with no anticipated need for treatment, at least over the next 6 months. The treatment study will enroll patients that are starting treatment with anti-VEGF for center-involved diabetic macular edema. And exclusion criteria include things like no previous treatment for DRD or diabetic macular edema, any substantial non-diabetic eye disease, and anything that really would prevent a participant from performing the study procedures or tests. So here's a snapshot, I'm not going to linger on it, of the study procedures that we have envisioned. The natural history study we anticipate will include something around 400 patients with a likely duration of about 4 years. We need long-term studies to really understand the evolution of these characteristics. The treatment study may include about 100 patients over the course of 1 year. On the left, you see a panel of the ways in which we typically characterize retinal disease in patients in our clinics: eye examination, best-corrected visual acuity, a range of imaging, including ultra-wide field color photos, fluorescent angiography, where we inject dye into a peripheral vein and watch as it transits through the retinal vessels to get a sense of abnormal vasculature or areas of leakage. OCT, which is a measure of retinal thickness, and OCT angiography, which gives us a high-resolution look at the central retinal vasculature. On the right, you'll see the procedures that we've identified as sort of new and promising for this study. And we've looked at both light-adapted in yellow and dark-adapted in blue testing so that we can identify changes that occur in both rod and cone pathways. My last 3 slides, I'm going to go through somewhat quickly. They just kind of highlight the tests that we're featuring in these that really rose to the top as we convened our groups on visual function over the last year. The first is the quantitative contrast sensitivity device, which can be done across sort of different lighting conditions. And the meaning of the deficits is really a look at general dysfunction of cells across the retina: rods, cones, retinal ganglion cells. We have data available for patients with diabetes with good evidence of good repeatability of tests from one to another, and also some understanding already cross-sectionally of how these tests change in patients with increasingly severe retinopathy. The testing time is about 4 minutes per eye, and the patient can do it without a chin rest or a head rest, with a fairly low burden on both the patient and the technician that's administering the test. The Retival ERG and pupil response device also became of high priority for us. This can measure functioning of bipolar cells and retinal ganglion cells, but the full field stimulus looks at overall retinal health. And there's been some really interesting 3-year longitudinal data that suggests that there's an 11-fold increase in the risk of need for treatment and an ocular intervention due to diabetic retinopathy with abnormalities on this test. Again, with some preliminary data already in terms of test-retest variability. And we think the testing time here is about 9 to 10 minutes for the light-adapted testing and the dark-adapted testing. There will be some time in between for patients to dark-adapt. Finally, the objective field analyzer is a perimetry, a visual field test that is objective because it looks at pupillary response rather than asking patients to indicate when they can see a stimulus. We have identified 2 protocols, the W20 and M18, which measure different regions of the retina in terms of response. There's data on test-retest reproducibility and cross-sectional data from 5 published studies with some longitudinal data as well. Again, relatively easy for the patients to accomplish with just over a minute in terms of testing both eyes. So I'm going to end there with just a look to our next steps. We will be forming a joint Mary Tyler Moore Vision Initiative and DRCR Retina Network Protocol Development Committee that will meet over the next month, and our goal is really to finalize these studies fairly quickly. There's been a lot of effort that's gone into their development already. So we'll be looking at questions at finalizing study design and feasibility, the statistical analysis plan, identifying our participating clinical sites, creating the web-based report forms for data collection, and so forth. We'll be looking to confirm our sources of support and collaborators as well, and the goal is to begin study recruitment perhaps in Q2 of next year. So we hope to have an update for you as we come back, hopefully next year around this time. All right, so I'd like to welcome an esteemed guest to the stage. Dr. Michael Chiang is the director of the National Eye Institute. We're very grateful to have him with us today, and we've asked him to talk about perspectives from the National Eye Institute and priorities in DRD research. Thanks, Mike.
Yeah, Jenny, thank you very much. I'm not so sure about the esteemed part, but I'm here. So Jenny, thanks. I'm really privileged to be able to talk about some of the things that we're interested in at the National Eye Institute. And I wanted to focus on a few topics, research, collaboration, and data science. And I just want to call out two people who are in the room now, Michelle Rybar and Carrie Getz. Can you raise your hands? I don't know where you are. Okay, in the back there, because they have a lot to do. If you're interested in some of these topics, Michelle and Carrie would be great contacts for you to talk with here about that. Okay. One of the challenges that we deal with in medicine is that it is subjective and qualitative. Okay, those of you who are not clinicians, who haven't seen these data, would be amazed how much discrepancy there can be when you get different experts to look at the exact same data. And I think that's kind of one of the premises of what Jenny is talking about today. And there's been a lot of work in AI and imaging to deal with these questions. I captured this tweet from Eric Topol a few years ago. Of all the medical specialties, most people think radiology is leading the AI movement, but it's actually ophthalmology. Okay, and there's several reasons for that, and one of them is because there's so much imaging data that's out there. About a month ago, David Huang, Jim Fujimoto, and Eric Swanson received the Lasker Award for basically developing OCT imaging. And then 2 weeks later, they were at the White House to receive the National Medal of Technology for the same thing. And it's technologies like that that are really driving advancement in artificial intelligence. Okay, so AI, imaging, and clinical data kind of go hand in hand. And we've gotten more and more this premise of the eye as a window to the, in this case, body. Because of these devices, there's such a literature now that's developing in this field of so-called oculomics. Basically, biomarkers that have been shown to have significance, and admittedly in somewhat preliminary studies, for diagnosis and prognosis of systemic diseases. And these range from neurodegenerative diseases, Alzheimer's, Parkinson's, to cardiac diseases, to psychiatric disease. And a week ago, the White House had a demo day called American Possibilities, where they highlighted technologies across the whole range, science, technology, energy, defense. And one of those technologies that was selected was OCT imaging. And so this is Carrie Getz presenting to an audience that included the president, Congress people. And so it was really big. This is a very, very exciting time for the vision community because of a convergence of all these technologies and the research possibilities. So this slide, I just want to talk about if things like machine learning are so good, why do doctors still have jobs in 2023? And I think some of this is probably obvious, but there's a couple of things I want to highlight. One of them is that you all know here that the first FDA-approved autonomous AI system in any field of medicine was for diabetic retinopathy. Okay, this is Michael Abramoff in Iowa. And that these systems are really, really good as the task narrows for what you're asking them to do. So Abramoff's system is really, really good. Does a patient have clinically significant referral warranted diabetic retinopathy or not? But if you give those systems a picture of a central vein occlusion here, they don't know what to do. And so there's a lot of these challenges with integrating multiple findings here that were really not there. And another big challenge is what I'd call generalizability and bias. Almost all clinical research studies are what I'd say, very homogeneous. And I used to work in Oregon, where the population is not very diverse. The real world, okay, there's multiple imaging devices, different races, different populations. And we worked on this study a few years ago, looking at this. And this is not the best study. It was not the first study in this area, but it happened to be one that I was involved with, so I know it the best. But the topic here is pediatric retinal disease diagnosis in babies. So it happens that here in the first row, if you train an AI system using North American data and test it in North American babies, it works really well. But if you train it in babies from Nepal and test it in babies from North America, it works really poorly. Okay, but if you build a data set of North American plus Nepalese babies, train a system, and then test it in either of those groups independently, it works really well. Okay, and so the take-home point from this is that this is a huge problem. And the power of healthcare data is really achieved through large, diverse, multi-centered data sets. Okay, so this is why I love, Jenny, the stuff that you talked about earlier. And so really, really important to keep in mind. So we need large AI-ready data sets to avoid these problems of bias. Now, I want to make sure you're aware of two different projects here that we've gotten really involved with at the NIH. One of them is called Bridge to AI. Okay, it's a trans-NIH project, any field of medicine, and it was $130 million over 4 years to build data sets. Okay. And of the 4 groups that were funded using this $130 million some dollars, one of them is led by a team of ophthalmologists, Aaron Lee and Cecilia Lee at the University of Washington. And that is a big, big win for the community here. It just goes to show how much work there is in this area. Now, another thing to make you aware of is a project called Aim Ahead. Okay, this is a $100 million project. Again, it's trans-NIH, any area of medicine. And the purpose here is using machine learning and artificial intelligence to address problems involving health disparities. And so, how do we do that? Build diverse data sets, train a diverse workforce, and develop AI approaches to detect bias. Okay, and I really hope the vision field can play a big role in this because photos are objective, okay, whereas a lot of bias comes from subjective things that end up in the medical record. Okay, so a point here is that we really care about this topic of building large data sets and diverse data sets. Now, one of the challenges, Robert mentioned data sharing in his introductory remarks. Most of us don't have $100 million okay, to build huge data sets. And so, we need data sharing and harmonization. And there's a paradigm in the world where often there are multiple what I'd call small studies that go on in parallel where people are trying to answer the exact same question. But all the studies are individually underpowered to really be able to answer that question and move the field forward. And so Martin Myers in his introductory talk mentioned this concept of the whole being greater than the sum of the parts. And we don't want an ecosystem where the whole is less than the sum of the parts. And so one of the challenges is that these different investigators are generally not incentivized to share their data. Okay, and the fact of the matter is that even if they did want to share, in general, you can't do it. When you don't collect data in a harmonized way, okay, you can't just combine your spreadsheets that way. These are huge problems. And so, how are we trying to deal with this? There is an NIH data sharing policy. Okay, and that's what I'd call a stick. Okay, if you don't, come up with a data sharing plan, we don't fund your research. And Maureen Maguire, Tom Gardner, they're on our council, and they can confirm to me that the community is not 100% thrilled with the data sharing plan. It's a lot of challenges with that, and so what I really think we need is carrots to go with those sticks. How can we truly incentivize people to want to share data? And about 2.5 years ago when I started this job, I actually got with Stephen McLeod and we talked, well, what can we do to bring the community together? And so basically I meet up with the editors-in-chief of the leading vision journals a few times a year. And we come up with, what can we do to try to drive the community forward? And one of the things that we did a few years ago is create a new publication type, a data set or software library. And so the idea is you write a paper about your data set. And it was our hope that this would become a thing within the vision community where people would then feel like you can get credit for publishing your data as an independent, standalone project. People could cite it, they could find it using PubMed. And the first journal that implemented this was TVST. Marco Zarbin was the editor-in-chief at the time. And I hope it's going to be things like that we work on together that really incentivize data sharing and team science. And the other thing that we lack is a common data model for ophthalmology. Okay, right now, clinical data, in different EHRs are kind of like apples and oranges. And who's working on this? A lot of work in collaboration with the NEI and the American Academy of Ophthalmology. It's being led by Sally Baxter, who's currently in San Diego, by Carrie and Michelle in the back there. Okay, and more about this coming up. So when it comes to sharing data, there's two approaches. Number one is a centralized approach, where everything comes into a common registry. And the American Academy of Ophthalmology's IRIS registry is one example of that. Michigan has a source registry in ophthalmology. And the pros here are that there's a lot of intuition, and there's some simplicity to pulling everything into a common repository. One of the cons is that it can require extensive data sharing requirements, and also with large data sets, images, it's hard to transfer them because the data are so big. Okay. Now, an alternative approach, and these are not mutually exclusive, of course, is a federated or distributed data network, where the data stays within the individual sites, and the analysis methods get shared, basically. And so a few examples of this, there's a European data network, the Eden network. There's a pediatric network, PedsNet, and there's an Odyssey network that I'll talk about. And here, the pros are that it's scalable, and you can support things like large imaging, okay, ocular data, systemic data. And of course, the cons are that you need to standardize. Okay, everybody still needs to speak the same language. And along those lines, interoperability is a huge challenge. Okay, we have terminologies, ICD-10, CPT, SNOMED, that are meant to sort of do that standardization. But EHRs in general always have some local or proprietary coding systems beyond that. And that makes it difficult to collaborate. Common data models, what they'll do is they'll map these coding systems in an overall sort of wrapper. And that really promotes federated data analysis. There's a lot of different common data models out there. I2B2, PicorNet, some of you are aware of. And Odyssey is the one that we've really gotten interested in. There's a common data model, OMOP, that we think has gained a lot of traction. Okay, it's used by projects like All of Us, the European Data Network. And what happens here is that all the data are concepts that are encoded in these usual terminologies, but there's this overall data model that overlies them. And when you have that, the top row of this picture represents different EHRs that all use their own ways of organizing data, but then you map them to a harmonized standard. And when you do that, you can then create standardized phenotypes, standardized cohorts, and then use federated tools to extract phenotype and basically analyze these data in a consistent manner. And so one of my aspirations is that the ophthalmology community would be able to do something like this and collaborate at scale. And one example of this, Cindy Tsai at Johns Hopkins did a really interesting pilot study involving OMOP, where this was trying to answer a simple question, the relationship between kidney failure and anti-VEGF injections. And in a very short amount of time, a couple months, she was able to gather a cohort involving 12 sites, totally federated data analysis, 400-some million patients, 6 million patients with blinding eye disease, and answer this question. Okay, and so I think that really gave a proof of concept of the power of this type of analysis when everybody speaks the same language. Okay, so my last anecdote here is ocular imaging standards. So much of our data is in images, okay, but the problem is that these are often locked in proprietary standards, and researchers and clinicians can't get access to that raw data. Okay, in the department that I used to work in, we actually maintained 2 image management systems because they couldn't talk to all the devices out there. Okay, this is very common. researchers can't get access to their raw data. And this really hasn't advanced significantly for the past 20 years, despite the fact that there are DICOM standards for every common ophthalmic imaging modality. And so the radiologists figured this out, almost 30 years ago, and we really haven't. And so when I started this position, we got together with folks from the FDA and the ONC, the Office of National Coordinator. The ONC manages electronic data exchange. The FDA manages, device regulation, and we manage research funding. And we thought, all of us felt that this cannot continue this way. Okay, it's not good for patients. It's not good for research. And 6 months, so we held a workshop. Carrie Getz was the person who led that workshop last year. It took a year to plan that workshop. 6 months after that, the AAO and ARVO published a policy statement saying that, that endorsed it. We can't continue that way. And there's a paper that's in press right now. What are the levers that we plan to implement to try to move toward an ecosystem that is more standards-based for ophthalmic imaging? And in short, I don't feel like I can, in good conscience, continue funding grants if they use devices that don't adhere to standards. And the question here is, how do we move to that system? Because I think that's how we're going to do the best clinical care and the best research. Okay, so. This is my last slide here. I think there's so much exciting stuff that is going to happen here today, involving data science, imaging, new sort of endpoints, standards, interoperability, data sharing, collaboration, and just really looking forward to what this group does in those areas. So thank you very much.
Thank you so much, Mike. So I'm going to invite our panel up to the stage. We're going to have about a half an hour of discussion just on the clinical research issues, the perspectives that Mike has raised from the NEI perspective. So I'll invite, in addition to Dr. Chang, Dr. Dolly Chang from Genentech, Maureen Maguire, Stela Vujosevic, and I'm also Ryan Barunas, who is subbing in for Sandra Puczynski, who unfortunately was unable to be here today with us in person, but I think is online. So, yes. Oh, we're going to, let me make a note that we're going to pass mics around. So please, we encourage questions from the audience. If you've got a question, just raise your hand. We'll either bring you a mic. There is, if you're feeling particularly athletic, a mic box that you can throw, but please try not to take anyone out with it as you do that. We don't want to lose anyone. And the mic is on the little X on the top, so we will be joyfully throwing that one around as well.
Just hold it like a normal mic. That down here, just like a normal mic.
It's foam. Yeah, it's not hard. And Tom's going to be moderating any questions that come in from the Zoom participants as well. So I'm going to start actually just going down the row, and we've got a very diverse group of panelists here with very different perspectives: federal, industry, clinical trials, retina specialists, patient advocates. From each of your perspectives, and Michael, start with you, can you comment on, since many of our clinical studies right now are looking to really develop new endpoints or validate new endpoints, is there a need, from your standpoint, in diabetic retinal disease for these endpoints?
Jenny, let me speak generally to this, because I'm not an expert in diabetic disease. But I guess I would say that one of the things that I think is, I've more and more realized, is that we're all on a spectrum, from complete health to complete illness. And the question is, where are you on that, and how do we get you more toward the complete health side? And I think that one of the things that I took away from listening to your remarks is that the endpoints that we use right now are things like, for example, based on fundus photography, or for structural endpoints, or based on visual acuity for functional endpoints. And I think what you're arguing is that those are somewhat primitive, if you will. They're either decades old or a century plus old. And if we can come up with new endpoints based on 21st century technologies that help us classify where are you on that map from complete health to complete illness. And if it does a better job of identifying where we are structurally and functionally, I think that would be a really, really interesting question, and it would hopefully benefit patients because we want people to get more to the health side.
Thanks, Mike. Stella?
Yeah, from the clinical standpoint, as a retina specialist, I think it would be really important in diabetic retinopathy to have new endpoints, because if we think only on visual acuity, we know that it is not the only one visual functional impairment or parameter that describes the impairment in our patients, and it is a very, I would say, late endpoint. So I think we would all like to treat our patients before, in order to, let's say, to prevent the visual function loss in the patients. So I think that would be really something crucial, also in order to have new treatments and earlier treatments, and the treatments not just for late-stage disease, such as DME or PDR, but also in, let's say, in the initial stages of the disease.
Terrific. Maureen?
Well, I don't know if I'll be able to elaborate too much on that, but I really do think that if we're going to prevent vision loss, if we're going to slow down progression, we need to be able to measure early types of progression, and right now we don't have the tools to do that.
Mm-hmm. And Dolly, from an industry perspective?
Yeah, sure. I think that, the two endpoints that has been served us really well is BCVA, the best correct visual acuity, and the DRSS score. But that only captures some part of the disease process. It's usually the later process, the angiogenesis portion, the macular edema portion. But we know that diabetes is a very complex disease. So, what about the neurodegeneration portion? What about the ischemia? What about the perfusion and all the other perspective of the disease? So I think that we really need other endpoints that we can capture that, and also at the same time that we can capture what the drug actually do to these disease process. So absolutely, with the new technology, we hope that we have more sensitive way to pick up the disease and also monitor the disease progression as well.
Terrific. And Ryan, you spoke really movingly about your experience and treatments this morning. Are there aspects of the patient experience that you'd like to see incorporated in our clinical measures of how patients feel, function, and survive?
Right. Well, to your points, earlier screening is so beneficial. I think that, despite the prescribed annual screenings I was undertaking, and then following my initial retinopathy, monthly screening, I still underwent that ordeal. And, obviously something was going on for probably years before that. And it was unnoticed, undetected. But, even going through the process, there was no clear message of here's what to expect. Here's what you're going to see on the other side. And I retained my core vision, my straight ahead vision, but I'm still, learning to adjust and adapt my life to having a narrowed field of vision, the floaters, glare at night. It affects my driving ability at night. It affects, my tennis game, my hockey game. It's, it's definitely something that patients learn to adapt with, but I think that wasn't very well communicated up front, and I definitely think that would help with the patient experience.
We need better ways to describe and set expectations, and then also set ways for patients to describe what they're seeing to us, maybe. Maureen, you've worked in a number of retinal disease types aside from diabetes, age-related macular degeneration, inherited retinal degenerations. Are there lessons in your works with other fields that we might be able to take into the DRD space?
I have two stories. One is a cautionary note, and I did most of my work in age-related macular degeneration, and most of the retina specialists in this room certainly know that the hallmark of early AMD is drusen. We also know as people get older, they get more drusen. We know that the more drusen you have, the higher your risk of neovascular disease and geographic atrophy. So these are all good things that would lead up to a surrogate marker. If we could reduce drusen, maybe we could reduce vision loss from late-stage disease. But we did do a series of trials looking at laser treatment to reduce the drusen load, thinking that that would slow down the disease process. And unfortunately, we were successful in reducing drusen, but had no impact. It's the only study I've done that had a p-value of 1.00. No impact on late disease. So this is a cautionary note that, yes, we need to develop sensitive markers, but we need to carry through and evaluate them for if we change the marker, do we change the clinical outcome? So that's the cautionary note. In the second story is in the area of inherited retinal degenerations, where I work with the Foundation Fighting Blindness Clinical Research Consortium. And we're looking at very rare diseases. A sample, I'm used to public health diseases, glaucoma, AMD, and now I'm in a room full of people where 100 patients is like huge. So it was a change. But we've done some studies with 100 patients on a specific disease causing a gene variant. And now we have 4 years of data. And what we're seeing is that the measures that we've used classically for glaucoma and a diabetic retinopathy, AMD, just are not sensitive to pick up progression on these slowly progressive but unrelenting retinal degenerations. So our group has come to the conclusion that we really need to use continuous measures, such as change in mean sensitivity over the visual field over time. That seems to pick up progression, but it's not something where somebody says, aha, yesterday I could see and today I cannot. It's a very gradual progression. So we need now to make sure that these are meaningful changes in visual field, but little changes accumulate over time. So I think that's what we're looking for, are the early changes that may move relatively slowly, but predict the future.
Thanks, Maureen. Questions from the audience? Excellent.
Yeah,
Dr. King.
Yeah, well, thank you for wonderful presentations, and George King from Boston. I also want to congratulate Michael and Jenny for a wonderful talk, and Robert as well. So since we're looking at biomarkers and for early stage of DR, from the discussion I heard, rightly so, mostly ocular-centric approach. Being an endocrinologist, I wonder what's the opinion of what is the role of family doctor, PCP, internal medicine doc, endocrinologist like myself. How could we follow, and besides DR, besides just telling patient to go see an ophthalmologist? Because for the renal disease, at least we have estimated GFR, creatinine, BUN, microalbuminuria. Some markers, they're not perfect. So what do you all think is the role of non-ophthalmologists in preparing or follow DR?
Yeah, George, thanks for that question. I think there's a huge role for primary care and non-ophthalmologists in managing this. In fact, one of the places that we started out here was talking about telehealth and sort of AI diagnosis for diabetic eye disease. And in fact, I would argue that if that gains more traction, then diabetes screening may shift more to a primary care. Currently, it's done by ophthalmologists and optometrists. And the more it's automated, the more it may shift to a primary care disease, where endocrinologists or pediatricians or internists are responsible for that diagnosis. So I think there's a lot of interdisciplinary work that has to be done in this area for exactly what, and that's a clinical thing. And from a research perspective, there's so many more avenues that I know that you've thought of a million times.
From a research perspective, I'll say that the ability to monitor risk of retinopathy or retinopathy severity from peripheral blood testing or other samples would be a huge boon to the community. I think there are a lot of barriers right now in placing cameras and technicians to take the images in primary care or endocrinology offices, whereas there used to doing blood tests. So George, I know you've worked on RBP3 as a potential marker, and that kind of test, if we can validate it. So one of the things we're building into these studies is the ability to biobank samples, because we're very interested in making these available for assays for things like RBP3, other potential targets that might help with screening or monitoring. And this will play very much into Patrice's next session about biobanking as well.
Maybe I can just add on from the research perspective. As we know that the eye is the window to the vasculature of the body, so, one of the efforts that we are working on is to correlate the ocular retinopathy to the kidney disease, for example. For natural history study, we sponsor natural history studies that take a kidney biopsy and then to see what are the changes in OCTA, for example, correlates with the kidney changes. So I just want to emphasize it's a systemic disease, so we need to look at beyond the eyes and also the body as well.
Great. I think Tunde's got the box.
Yes. I've got the box, but they weren't game enough to throw it at me because there's a cup of coffee in front of me.
Speak loudly, though, Tunde, so we can hear you.
So they weren't game enough to throw it because there's a cup of coffee in front of me. Okay. So I wanted to say that I think in answer to your question, I think we are slightly our victims our own success. We were so successful of treating eye disease in the UK doing the screening that we sort of slightly taken out the eye out of the body. So part of our desire here will have to be to put it back and do some very good structured data together with the diabetologists. But if we go back to the original question as to what you could do, so on the UK screening data, we looked at just the images, and if you just look at the screening results, and if you know who has changed from 1 year having no diabetic retinopathy in both eyes to having diabetic retinopathy in both eyes, so it's from nothing to something, can be just one microaneurysm in either eye, your chances go from 1 in 20 to have significant visually threatening disease in 4 years to 1 in 3. So if you get the results as a primary care physician, if the primary care physicians get the results of the screening, and they just have a look if there is a change, the people that you really need to take care of are the patients who went from nothing to something, because they go from 1 in 20 to 1 in 10 if it's one eye, and 1 in 3 if it's both eyes in 1 year. So those are your high-risk patients, which might actually drive some of the protocol thinking as well, because we can actually really reduce the number of people needed in trials if we look at the last change before baseline, for example. And that's big data because we did it on most of the UK screening protocol.
Terrific. Thank you, Tunde. I think there's a comment from Ramin right behind you.
I have a question. So one of the problems is that for visual acuity, for example, there is a real impact in the life of the patient, which is partly due to convention. So if it's below some level, you cannot drive. But this is because it's accepted like this. But we have nothing really connecting other endpoints, like let's say contrast sensitivity or small scotomas, to anything in real life. We don't know what is the impact, and we don't know how have it. If a patient has zero contrast, which could not happen, but let's imagine, he can still drive, for example. So how we can manage to make the link between the new endpoints that we are going to create this with these patients with something real in their life, so they're valued in terms of impact of human suffering or money or whatever, to have reimbursement, to have covering, to have all the discussion, people to invest in this.
It's a great question, Ramin, and I suspect that this conversation will continue into the regulatory panel that will be a little later this afternoon. I'll note, so Sandra Puczynski, who was going to join us and couldn't be here, wrote notes to me about, in terms of critical aspects of the patient functional experience for us to capture, about things like pain and anxiety related to treatments, fear of vision loss, loss of independence due to vision loss, the idea that treatments for DRD can be very difficult and can result in both loss and hopeless feelings for patients in a vacuum of dispassionate care sometimes. So these all speak to how patients feel, certainly, and your questions about how patients function in ways that we don't currently capture is important. Stela, you've led our working group in terms of quality of life, and we've had a lot of discussions about PROs, and maybe you and then Mike can comment on sort of efforts in those areas.
Yep. We did this narrative review on the patient-reported outcome measures and the quality of life that were used in diabetic retinal disease, together with Emily Chu, Shobha Sivaprasad, Lynn Labriola, and Ekos Lamorex. And actually what we found is that we don't have, let's say, sensitive prompts for the early stages of the disease. We just have the prompts that can detect, let's say, the changes at the very late stages of the disease. For example, when we have DME, okay, and we have the visual impairment. So the problem is that we really need, let's say, our new prompts, or to extend or modify those that are already available, in a sense that they can capture these early stages of the disease, and also that they can capture longitudinally the changes in the severity of the disease when, for example, we treat the patients. We don't have these prompts in these, let's say, large longitudinal studies. Then obviously it would be important that these new prompts could be digitally administered, not only paper-pencil administration. That's something really important, and obviously to be freely available. So definitely there is still, I would say, a lot of work to do from that point of view, and it would be really important to correlate these new prompts with some other aspects of visual function, not just visual acuity. So some other aspects that are more precocious, that we then need to validate before, no, in order to use them in the clinical practice, in the trials, as the endpoint, or whatever.
Terrific. Mike, NEI just held a workshop on this subject. Comments from that perspective?
Yeah, Jenny, maybe I'll just follow on what Stella said by saying why we held that workshop. When I started about 3 years ago, one of the first things we did is looked at our mission statement, and we spent 6 months revising it. And so 6 months for one line, of text that said our mission is to eliminate vision loss and improve quality of life. And I think the challenge is that we don't have a very good way of measuring quality of life. And in a nutshell, that was the reason that we wanted to, hold this workshop. And we really means us together with the FDA. And there were a couple other groups that were really closely involved in planning it. PCORI, the American Academy of Ophthalmology, and also FFB, Foundation Fighting Blindness. And my hope is that we can develop a metric that measures vision-related quality of life that's endorsed, for example, by the FDA, where you can just, it works for everybody the way vision acuity works for everyone. And that also there may be a battery of linked PROs that would be used for research studies. And that would be my vision for this, but we got to leave it to the experts for what, what's going to come of it.
Terrific. Thanks. Other comments, Maureen, Dolly?
I think that from drug development standpoint, the PRO portion, it's really hard to capture what the drug actually does to the pathway. So, we often administer VFQ-25, and there are many domains that are captured, the things that beyond just what vision impact. So I'll be very curious about regulatory perspective, for example, how we can do better in terms of PRO, and then see how we are going to measure how the drug impact the patient's quality of life and function.
Terrific. I'm going to give Ryan the last word on this panel, really by asking Ryan, as we do all these clinical studies, and there are a lot that need to be done, any advice on how we best keep patients front and center in the effort to validate new endpoints and for regulatory trials?
Right. Well, as I mentioned, the after effects of all this, I retained my central vision, but I think that ideally I'd be able to still maintain my peripheral vision. I'd be able to have that sense that I'm going to have independence in the future. I'm terrified of reoccurrence. I know I had a successful outcome the first go around, but there's no guarantee that I won't. I hope I don't have to go through this again, but the second go around might not be as fortunate. There's a lot of damage. Every time I go to my ophthalmologist and the retina specialist, they remark at how damaged my eyes are and how fortunate I am. And they remark on the scar tissue that remains, and that more than likely in the next 5 to 10 years, I will have cataracts that will need to be removed. So I think that just having that hanging over my head, it's, as you mentioned, Sandy's remarks, there's the anxiety and the stress that goes along with this as well. And, as I mentioned as well, I think communicating to the patient exactly what to expect, what the steps are, and what the expected outcome are would help alleviate some of that. But hopefully we can get those addressed as well.
Terrific. I'd like to thank the whole panel. Thanks to the audience for the questions and great discussion. And I think Doreen has a few announcements before we go to break.